place for me to also put in next week, because of Veterans Day on Tuesday, there is no lecture or labs, okay, for this class. Don't know about your other classes, but for this class. Then we're going to come back. We will have an exercise lecture, and then the labs will meet. And then I believe after that, we have no lecture or labs because of Thanksgiving. And then we're going to come back and have a lecture and lab on the kidneys, and that will be our last lecture and lab. And the week after that will be the final. So the final is coming up pretty quick. This is one of the reasons I don't like the fall semester, because it gets broken up a lot, not only because of Thanksgiving, but because of our roaming Veterans Day holiday. Okay? So just make sure everyone understands that. Make sure and follow what I have in the syllabus. I think what I have on the syllabus is what I've just said. If it's different, follow the syllabus. Okay? Okay, so returning to the subject today, we're going to talk about a little bit about lung anatomy, but we're going to also talk about respiration. Respiration encompasses 3 different functions. There is ventilation, and ventilation is the movement of air in and out of the lungs for gas exchange. The second function is going to be gas exchange, and we actually have 2 sites for gas exchange. In the lung itself, we're going to have what's called external respiration, because we're having gases exchange with the alveolar sacs and the plasma. But then when we get the blood to the tissues, we have, oops, where did that go? We have our second respiration site. That's called internal respiration, and that's where gases are going to go from the plasma into the actual tissues. O2 utilization is simply how we use oxygen in the cells, and the primary function of oxygen is to help create ATP. Gas exchange is always going to happen via a passive process, via diffusion. Okay? Now, I do show this slide. It's kind of the only one that I have that shows the complete respiratory system. Ignore what's at the top. We are going to divide the respiratory system into the conductive zone and the respiratory zone. That's the way we teach it in anatomy. I will tell you, however, that some people do talk about upper and lower respiratory systems, but not in here. The thing that I like to show about this is that in the respiratory system, we have a series of passageways. We start with the nasal, or it could be the buccal cavity, which is your mouth. That's going to go to the pharynx. From the pharynx, we divert gases forward through the larynx, where you have your vocal cords. Then it goes down to the trachea, and it will go out through the bronchi and eventually go to bronchiolar passageways, and then it ends in the alveolar sacs. One thing I want to point out is that this is a one-way system. Gases move into the lungs and then move out through the exact same passageways, okay? So it's not like our circulatory system where blood leaves the left side of the heart, comes back to the right side, and we pump it through the body. So we're going to look first at the conducting zone. The conducting zone, first off, I will tell you, the conducting zone ends where cartilage ends. So that is really anatomically what divides the conducting zone into the respiratory zone. And there are 3 important functions in the conducting zone. They're listed on this slide. The first one is that we need to warm the inspired air. Typically, our body is warmer than our climate. And what you have to think about is that the air coming into the lungs is going to be exposed to your entire blood volume every minute of the day. And so if you had cold air, you would actually chill yourself off very quickly. So we are going to warm the air to body temperature. Now, there is more of a problem when you go out, say, to the desert. You can't cool the air off coming into the lungs very effectively. And that's why in the desert, you're going to use thermal regulation through sweating to cool the body down. The other thing that we have to do is that, and we'll talk about it later, your body is always producing or consuming extra water. And so in a controlled manner, we are going to humidify the air going down to the lungs. In the same manner as we don't want to chill the blood in our body, if you had a lot of dry air going down into the lungs, water would move through osmosis and leave the plasma. And it's estimated that if we didn't humidify the air in the conducting zone, you might desiccate your body in a few minutes. So we make sure that it's the same osmolarity so that we have the same water content in the conducting zone, so when it finally gets into the alveolar sacs, there is no osmosis going on. And the last thing that's important in the conducting zone is we clean the air. Okay? Whether we like to think about it or not, every time you take a breath in, you are breathing in bacteria, fungus, mold, some other microbes as well could be floating in the air. And to help to protect our body from this, we are going to secrete mucins along the conducting zone. Mucins are simply types of carbohydrate. We actually add water to it so that you have just a very thin layer of sticky substance. You could kind of think of it as a flypaper, okay? So now things can attract to that mucin layer, and we have all the cilia in the conducting zone. It's always taking mucins and anything trapped in it back up, where often we will swallow it and not even be aware of it, and then those microbes will go down into the stomach where we have hydrochloric acid, and it destroys them, okay? So we're going to clean the inspired air through the mucin that lines the conducting zone. When we go down into the respiratory or respiratory zone, again, the definition from this is the areas where there is no longer any cartilage. This is where we do see smooth muscle, and we see most of the area is going to be surrounded by capillary beds. So the alveolar sacs make up the majority of the respiratory zone, and they are completely surrounded by the capillary beds. This is where external respiration occurs. So you're going to get movement of gases from the alveolar sacs into the plasma of the blood in the capillary beds. Now, if we look at this area a little bit more closely, what I have on the left side is an actual micrograph of a high magnification of a capillary. Where's my pointer? Oops, sorry. This is the capillary here. This is the nucleus of the endothelial cell that makes up the capillary. This is actually plasma, and in the process of fixing it to make a slide, some of the contents of the plasma crystallized. And this is a cross-section of a red blood cell going through the capillary. On either side, we have alveolar sacs, okay? This is where the air is coming in and leaving the lungs for gas exchange. Now, one of the things that I want you to think about, and we always kind of go back to Fick's law in various aspects in physiology, the diffusion between the alveolar sac and the plasma, this distance is very, very small. And so diffusion is almost instantaneous. So when you're bringing oxygen in, it's going to diffuse easily to the plasma. And when we're bringing CO2, we're going to have a concentration gradient so that it can very rapidly diffuse out of the plasma. Now, on the right side of the slide, there's our cartoon version of the lungs. And the alveoli are going to be made up of what are called type 1 alveolar cells. We also call them type 1 pneumocytes, okay? They make up the structure that is the round alveoli. And inside, we also have type 2 alveolar cells, also known as type 2 pneumocytes. And that's this kind of strange-looking cell up here. And we also have resident macrophages. Those macrophages are there so that if any microbe does get through the conducting zone, we have something ready to phagocytose it and remove it before it can actually get into the body. Around the alveoli, we have lots of elastin. So this area on the outside, right in here, is made up primarily of elastin, and that's a very stretchy substance. The best analogy is like a spandex or a lycra, where it will stretch out easily and recoil easily. And then we have, right here, we have our capillaries in here, okay? Now, if you were to take all of these alveoli and put them together and stretch them out, they would cover a tennis court. So this is the type of surface area we have for rapid gas exchange, okay? All right, so now when we think about breathing or ventilation, this is going to happen in the thoracic cavity. And we have an abdominal pelvic cavity in the body that is divided into two parts by the diaphragm. Above the diaphragm is the thoracic cavity, and that is where you're going to have the lungs, the heart, large blood vessels, and in a very young child, a thymus. Below the diaphragm is the peritoneal cavity. That's where you have the digestive system, the urinary system, and reproductive systems, okay? When we look in the thoracic cavity, the main organs are going to be the heart and the two sets of lung lobes, okay? All of these organs are surrounded by serous membranes. Now, serous membranes are very thin membranes that wrap around organs, and they have a very small amount of fluid between them. We call the serous membranes by the organs that they wrap around. So there is the pericardial serous membranes around the heart, and then we have pleural membranes around the lungs. These membranes, we also distinguish the side that is touching an organ from the side that is not touching the organ. And in the case of the lungs, the visceral pleura, our visceral is always going to touch an organ. So the visceral pleura is against the lung material, and the parietal pleura is going to be the part of the pleural membrane that physically attaches to the thoracic cavity wall. Okay? In the lower system, or in the lower pictures, we kind of think of pleural membranes as a balloon filled with a very small amount of fluid that's tied off and then wraps around an organ. So it is intact, and I will tell you that the pleural membranes are absolutely critical for breathing. They are going to be what helps to keep the lungs inflated. Okay? With respect to this idea and why they keep the lungs inflated, there's an interesting embryonic history. All of these structures in an early embryo are present. You have the thoracic cavity, you have the diaphragm, you have what are called lung buds, and you also have the pleural membrane, you have the heart and the pericardial sac as well. In the very early embryo, and actually through the embryonic life of a human, they don't need to breathe. They don't need to use their lungs because their gas exchange is coming from the placenta and the mother's body. So what's interesting is that as a fetus starts to grow, everything's growing proportionally except the lungs. The lungs are delayed, their development is delayed a little bit, although it does start to grow, but they never grow to the same extent as the thoracic cavity and the diaphragm. But the membrane, the pleural membrane, is intact from early on. And because of this unequal growth, you get a negative pressure inside in what we call the intrapleural space. Now, that intrapleural space is going to help to keep the very stretchy lung material expanded. And also, while you breathe, if your diaphragm or your thoracic cavity moves out, the pressure inside becomes a little bit more negative, which is then just going to gently stretch the lung material and have it go out and conform to the structure of the parietal pleura. When you allow the diaphragm to relax or the thoracic cavity compresses, then that very stretchy lung material will go back to the way it should be from the original position. It is important to have this kind of both push and pull. The lung is always trying to collapse the outward pressure to keep the lungs inflated. Now, this describes something that we call a pneumothorax, okay? So if somebody has a knife wound or a bullet wound, or something that is allowing air into that intrapleural space, then you have destroyed that pull of the thoracic cavity wall and the diaphragm. And the lung, the stretchy material of the lung, just collapses. Now, when the lung collapses, the proper term is something called atelectasis, and pneumothorax is just one reason for atelectasis, okay? When you have to reinflate the lung, you're going to have to seal up that the parietal pleura, and then suck the air out, but it doesn't take a lot of pressure to suck it back out to reinflate the lungs. So we know that both the visceral and parietal pleura are essential for breathing because they're going to normally help the lungs stay inflated and also help to stretch it out and then allow for the elastic recoil of the lungs when we breathe out. Okay, any questions about that? Okay, so we're going to talk a little bit about ventilation now. And Robert Boyle was somebody who was very interested in gases in general. And interested in gases, he was also interested in how we breathe. But of course, I think we all know that gases are a little bit difficult to work with. It's not like you can measure a concentration of a gas. You can't just scoop up oxygen from the air and figure out, well, how many moles of oxygen do I have? And what Boyle did was he actually sealed gases in a container, and that's what's shown on the left side of the slide. And he realized that in that sealed container, he could measure a pressure. And the pressure is a result because these gas molecules are diffusing very rapidly, and they'll hit the side of the container and sometimes hit one another. And every time there's a collision, a little bit of energy is released, and that energy is what we can measure as pressure. Well, then he made another interesting observation, which is shown in this slide. If he takes that sealed container, and it has a seal but a plunger, and he decreased the volume, he noticed that the decreased volume, those same gases had a higher pressure. And he started thinking about it, and one of the things that we can kind of imagine is that these pressures are somewhat representative of concentrations. If you think about it over here, if we have 5 gas molecules in 1 liter, and they are producing 100 millimeters of pressure, it's telling us that this is less concentrated than these 5 gas molecules in a lower volume that are producing 200 millimolar of mercury of pressure. So we can kind of view pressure as the concentration of a gas. Now, I will tell you that pressures are a little bit more complex. Pressures are equivalent if you're going from gas to gas, but they are a little bit different if you go from gas to fluid. But nonetheless, gases will always move down their pressure concentration gradient. So if these were 2 different containers, whoops, what did I do there? Come on. Where's my, there we go. If these were 2 different containers with gases, and if you hook the hose from the left one to the right one, gases would move from the left to the right. They would go down their concentration gradient. And that's true for whether they're both gases or from gas to liquid. Any pressure gradient, they will go down, okay? Okay, so So when we think about ventilation, and these passageways have no one-way valves, if we look at somebody who has not taken a breath in, the diaphragm is resting in its up position. It's a dome shape. So there is a certain volume of gases in here, and this volume and the pressure in here equals the outside atmosphere. When we then take a breath in and the diaphragm contracts, what we have done is we have increased the volume of the thoracic cavity and the volume of the space in the lungs. Well, that means that there's less gas molecules in here compared to the outside. There would be less pressure, so it would be negative pressure, and that draws gases back in. Once you have filled up the lungs with pressure, or the gas or the pressure, and it's equal to the outside, if I go back, and now I relax the diaphragm, I have all these extra gas molecules in here that are creating a positive pressure, and they are driving gas back out. So because of Boyle's law, we are able to ventilate. We can bring gas in and move gas back out, okay? All right. This is just to show you an example of this. This is an individual who has fully expired, which means they fully breathed out. What you see here, this is the liver. This is the outline of their heart. And you can see these white wispy lines are the ribs. And the dark area is the spaces in the lung. When the same individual, and they took an x-ray of this. When the same individual takes as deep a breath as possible, they took another x-ray. Now you can see that the diaphragm and the liver are now pulled down here. You see a very nice outline of the heart. And this was the original space, but now you've increased the space in the lung cavity, and this is what's allowing gases to move into the lungs. And then they would go back to this, and you would breathe out. Okay? So this is what is involved in ventilation, getting gases in and out of the lungs. Okay? Now to do this, of course, we're going to use muscles. And we have two sets of muscles. We have our inspiratory muscles, and the main muscle in the inspiratory muscle is the diaphragm. When it contracts, it pulls down and flattens out. But we also have external intercostals and scalenes and sternocleidomastoids. If you activate all of those, you will take the deepest breath in as possible. Okay? If you guys remember, those who've had anatomy, external intercostals, external muscles, you can think of putting your hands in your outside pockets, and they have their origin on the upper rib, their insertion on the lower rib. So when they pull up, they're actually pulling and ratcheting the ribs upward. If you have sternocleidomastoid and scalenes contracting, they will also help to pull the ribs up and forward while the diaphragm is pulling down. So collectively, if all of those muscles are working, you're going to get the largest volume change and increase in the thoracic cavity and within the lung. Anytime muscles are contracted, that is an active process. If you simply allow those muscles to relax, that is a passive process, but you will breathe out. Now, if you really want to breathe out as much as possible, we also have internal intercostals. And internal intercostals, if they go like this, you now have your origins on the lower rib, the insertion are on the upper rib, and when they pull down, they're going to pull down and in. So they're going to help to decrease the volume of the lungs, but we also can use the abdominal muscles. Now, the reason that they would help to expire is that when they contract, they're contracting around all those organs in the peritoneal cavity. Those organs cannot go out the back. We have too much fascia, too much muscle in the back, so they're going to go up into the diaphragm and push the diaphragm even further up into the thoracic cavity. And if you think about it, if I'm breathing in and I'm breathing in as much as possible, I'm going, and my lungs come out, everything up here tends to lift, the diaphragm's going to be compressing, and then when I blow out as much as possible, I'm going to go, and you can see the bend over, that is the abdominal muscles that are compressing, and all of the organs down below are going to go up into the diaphragm. So these muscles are going to help with inspiration and expiration separately. Again, the only time expiration is active is if you activate the internal intercostals and abdominal muscles. Okay, we're going to move on and talk about factors that influence our ability to ventilate. And there are three factors. We have compliance of the lungs, elasticity of the lungs, and something called surface tension. The only thing I want to talk about compliance and elasticity is that often people think, well, aren't they kind of the same thing? Compliance is simply an ability to stretch out. Elasticity is an ability to recoil, okay? I think the easiest way to understand the distinction between the two is think about a new pair of gym shorts. When you get a new pair of gym shorts, that elastic band, boy, it doesn't really want to stretch out. You've got to really pull it, and then you go and it snaps against your waist. At that time, you could say that that elastic band is not very compliant, but it has a lot of elasticity. As time goes on, you may have worn these for years, and now, boy, they just stretch out, and it doesn't maybe snap as well, so you need the drawstring to actually cause that waistband to be against your waist, okay? And this gives you an idea of sort of the difference between compliance and elasticity. Normal lungs shouldn't lose compliance or elasticity, but certain diseases will affect specifically compliance or specifically elasticity. That's why we think of them as separate, because we can destroy compliance of the lungs, for instance, with emphysema, or we can destroy elasticity with respiratory distress syndrome. The third thing that will affect ventilation is going to be surface tension present in the lungs. Now, I will tell you there are two locations of water in the lungs. We have about 3 mils of water in the pleural sac, in the serous membrane. We're not talking about that right here. We also have the water that is in the alveolar sacs that is part of that humidity, making sure that we don't have osmosis. And because we do absorb and secrete some water, especially in the conductive zone, we have a thin layer of water on the alveolar surface. This creates the surface tension. And that's because water molecules are attracted to other water molecules. To give you an idea of this, I've drawn this. So if you have one layer of water molecules, here we have water molecules that are hydrogen and oxygen bonding. Hydrogen and oxygen bonds make and break all the time. So what's interesting about this is as they make and break, water is going to tend to try and get on top of each other and actually create a smaller sphere, okay? So there is an inward pressure to cause a sphere like this to collapse. Well, some time ago, there was a French physicist named Laplace, and he was interested in this surface tension. And not only because of in the lungs, but he was also interested in blowing bubbles. And you know, if you blow a bubble, and one of those small soap bubbles, it goes and goes, and then it pops. Well, it's not actually popping out. It's compressing. And then, if you guys have heard of equal and opposite reaction, when it implodes, everything comes back out. That's why it seems like it's exploding. In any case, what he started thinking about and created what is called the law of Laplace is that this internal pressure is going to be equal to 2 times the surface area divided by the radius, okay? And so he just kind of thought about 2 different spheres. In this case, we're looking at 2 different alveoli, where this larger one has a radius of 2, the smaller one has a radius of 1. And if you simply replace the radius in this equation, the larger one, you have the pressure, the internal pressure is 2 times the surface tension divided by 2, so pressure will equal surface tension. In the smaller one, the pressure is 2 times the surface tension divided by 1, and that reduces down to the internal or inward pressure is 2 times the surface tension, okay? So we don't have to actually know what the surface tension is, but we know that in a smaller alveoli, because of that surface tension, it's going to have more force trying to make it collapse than the larger one. Well, when they realized that, they started thinking, wait a minute, how does that apply to the lungs? Because we are gently pulling out the lungs, expanding them, and gently allowing them to collapse. So that means that throughout the lung tissue, alveoli, some are large, some are small, some are intermediate, they're changing sizes as we expand and collapse the lung. And they started thinking, well, then why doesn't the small ones just collapse and add pressure to the larger and larger? And eventually you would just have 2 cavities, which would not be good for gas exchange because there wouldn't be enough surface area. So this is where they started looking at the lungs again, and they started to realize what was important about those type 2 pneumocytes. The type 2 pneumocytes produce something that is called surfactant. And I kid you not, this is the historical derivation of surfactant. It comes from surface-acting agents. You got to tell me whether people were high back then naming things, okay? I don't know if I could look at that and get surfactant out of surface-acting agents. Anyway, what they are secreting are a compound of phospholipids. Phospholipids have the glycerol backbone, a phosphate polar head group, and 2 lipid tails. And so they're putting these out amongst the water molecules. So we can envision this now, if on the left side, that would be an alveoli with no surfactant. Water molecules, as they compress, or as the alveoli shrinks or compresses, they're just going to keep bounding and making a lot of pressure that's going to try to collapse it. But now if you put surfactant in between the water molecules, it counters that. It prevents the water molecules from interacting with one another. And so we'll be able to have alveoli of all different sizes, and we won't collapse the lungs. Now, this is one of the things, surfactant is usually not made in the fetal lung until about week 35 in the pregnancy. And so when you have premature babies, we actually spray a soapy substance into their lungs, which mimics surfactant until their type 2 pneumocytes start making the natural surfactant. So this is what surfactant does. It's going to actually increase compliance instead of decreasing it if there was no surfactant present. So now what I want to do is move on to talk a little bit about how we test lung function. And the main thing that we test for are ventilation problems, okay? This is based on what we call spirometry. This image shows how we used to do spirometry. We would have a big tank of water and a big tank of just atmospheric gases inverted over it, and a tube that went up into the gases. And a person would close their nose off and breathe only in and out of their mouth. By breathing out, we would breathe air into that one tank way off onto the side here. And because you had more air in here, this container could rise. And then when you breathed air in, and this tube is way up in here, you would just breathe air in, and this would sink based on the amount of gas that you breathed in. They would hook this up to a roll of paper, and they were able to record the volumes of gases going in and out of the lungs. Now today, we actually use computers, same kind of setup where you have a nose clip on, and you breathe through a mouthpiece, but the velocity of the air is used to calculate the actual volumes that somebody is breathing in and out. When we look at spirometry, there's a couple things, and I do want you to know the terms and these values on this slide. Normally, what we call tidal volume is what you're doing right now. You're breathing in and out, and when you breathe in, it's about 500 mils. When you breathe out, it's about 500 mils. If you used the inspiratory reserve volume, so if you use the scalene sternocleidomastoids and external intercostals, you would get the inspiratory reserve volume, which is about 3,100 mils. And if you use the abdominal and internal intercostals, you would get your expiratory reserve volume, which is about 1,200 mils. Collectively, all of these added together are called the vital capacity. That means that on average, we should be able to take in 4.8 liters and breathe out 4.8 liters if we're breathing in as hard as possible and blowing out as hard as possible. The alveolar sacs should never completely empty. And so we do have a residual volume in the lungs. This is typically about 1,200 mils. And so to get the total lung capacity, which is the total amount of gas in the lung at the deepest inspiration possible, you actually add the vital capacity and the residual volume, you get about 6 liters. So at the deepest breath possible, we should all have about 6 liters in our lungs. Now, I'll also point out, in the conducting zone, there are gases that should remain there all the time. They make up what we call the anatomical dead space, or just the dead space. These gases are not available for gas exchange, and it makes up about a total of 150 mil out of that residual volume. Okay, so in talking about diseases of the lung, there are 2 major types, okay? We have restrictive diseases, and these are where the lungs have lost compliance, okay? The main type of diseases these fall into are pulmonary fibrosis, and pulmonary fibrosis is something that a lot of people get after COVID. You get scar tissue in your lung, and if you think about a scar or a scab, it's very stiff. Well, if you have a lot of scar tissue in the lung, it's not going to stretch out as easily. It gets a little scarred, and it's a little bit harder to stretch out. That is actually causing the restrictive disease. The other one is respiratory distress syndromes, and that's where either a premature baby who doesn't have surfactant or adults can have certain diseases that decrease the production of surfactant, hopefully just a transient condition. And that's adult RDS, okay? In these restrictive diseases, because it's decreased lung compliance, the lung doesn't stretch out easily on its own, so there is a reduced tidal volume, there's reduced vital capacity, but you can force it. If you use more muscles than you normally would, you should be able to stretch the lungs out and get the volume in. The other type of diseases are what we call obstructive diseases. And these are a more wide variety of diseases. They include emphysema, asthma, and pulmonary edema. And this is where we get the term COPD. C stands for chronic, meaning it's ongoing throughout the life of a person, and they're obstructive pulmonary diseases. Now, these are very different in nature, but they do all have one thing in common, and that is increased airway resistance. So just like if you have a constricted blood vessel, there's more resistance to blood moving through that vessel. Well, in COPDs, there is something that is causing more resistance to airflow. Because inhalation is active, you don't normally see it on inhalation. But because normally exhalation is passive, you see that people have more problem breathing out, and they generally have to force it out, okay? So that's the difference between the 2 types of pulmonary diseases, okay? Now, in spirometry, we do have a technique called an FEV1, which stands for Forced Expiratory Volume 1, that can help to detect if somebody has a COPD. And when you look at this slide, you kind of read it backwards. So you have somebody take a couple of normal tidal volumes in, tell them to take the deepest breath possible, hold it for a second, and then breathe out as quickly as possible. When somebody has normal lung function, they should be able to get most of this expired air out in the first second. And that's why this is called FEV1, because you're looking at 1 second after expiration. If you only get maybe 60 or less % of your total volume out in that first second, that is indicative of a type of COPD. We don't know which one, but that would kind of implicate that there is a COPD disease present. Okay, so now I'm going to move on, the last subject. We're going to start talking about gas exchange. And in thinking about gas exchange, we have to turn to another aspect of physics. We're going to look at partial pressures of gas and Dalton's law. In our atmosphere, we have oxygen, CO2, nitrogen, a couple of other trace gases. And we could measure the total pressure from the atmosphere, but each gas is contributing part of that pressure. And so the part that is contributed by a particular gas is the partial pressure of that gas. Now, Dalton realized, okay, I could just add up the partial pressure of oxygen, the partial pressure of nitrogen, partial pressure of CO2. When I add them all together, I get the total pressure. And so Dalton's law basically states that the total pressure of a gas is a sum of all the partial pressures. So you can see down here, if we have the atmospheric pressure, it is comprised of the partial pressures of every gas. And at sea level, atmospheric pressure, 1 atmosphere is 760 millimeters of mercury. So why is this important? gases move down their pressure gradient. It doesn't matter if you have 760 millimeters of mercury of pressure for oxygen exchange. What matters is the pressure of oxygen going into the plasma. So what we're going to be concerned with is the partial pressure of the gases that have to exchange throughout our body. And so as an example, most of our atmosphere is made up of nitrogen. About 79% is nitrogen, about 21% is oxygen. And if we know that we have an atmospheric pressure of 760 millimeters of mercury of pressure, you multiply this by the percentage, and that means that nitrogen its partial pressure is 600 millimeters of mercury, and the partial pressure of oxygen is 160 millimeters of mercury. This is what's going to be important at understanding oxygen movement in and out of our bodies, okay? So when we look at the lungs, we know that in the lungs, we have a partial pressure of oxygen of about 100 millimeters of mercury. And that's because when we breathe in, there is still leftover gases, leftover CO2 and oxygen, so it gets diluted a little bit. We also know that the partial pressure of CO2 is about 40 millimeters of mercury. These will equilibrate very quickly so that in our arterial blood, we should have those partial pressures going down to the tissues, where we're going to then have oxygen diffuse into cells to be used to make ATP, and CO2 will start diffusing out as we make it, and then we're going to go with a partial pressure of oxygen of 40 millimeters of mercury and a partial pressure of CO2 at 46 back to the lungs, and we pick up oxygen and get rid of CO2. Okay? I will just point out here, oxygen varies from a partial pressure of 100 millimeters of mercury in the lungs to less than or equal to 40 millimeters of mercury in the tissue. CO2 varies from 40 millimeters of mercury in the lungs to 46 millimeters of mercury in the tissues. And I am going to stop there, and I will write up a supplement for the rest of the lecture today.